The regulation of eIF4F complex signaling is associated with increased translation of mRNA subsets that are involved in cancer proliferation and survival. Here we describe an eIF4E-eIF4G cell-based protein-protein interaction assays that enables us to assess drug-induced perturbation in eIF4F complex integrity in live cells. There is a growing interest in the development of modalities that efficiently target protein-protein interface. We envision that our eIF4E-eIF4G PPI assays will help in fostering these strategies and validate them. This assay would provide an optimal primary screen for it to lead optimization of eIF4E-eIF4G inhibitors. Seeding the cell correctly and performing the transfection are the most challenging aspect of this protocol, since they may reflect directly on the expression of the PPI reporter system.
After thawing and counting cells, seed 6 well plates with HEC293 cells, using 2 milliliters of standard growth medium per well. On the morning of day 2, for each planned transfection, dilute 9 microliters of liposome-based solution in a tube containing 125 microliters of reduced serum medium without phenol red. While the diluted liposomes incubate at room temperature for 5 minutes, prepare the master mix of DNA by diluting 3 micrograms of each plasmid in 125 microliters of reduced serum medium for each transfection tube. Add 12 microliters of enhancer reagents